A very good morning to all of you. My name is Sara Iqbal, and I welcome you all once again to the annual Fellows meeting. We are going to start the meeting with talks by India Alliance Fellows. They're going to talk about research questions that keep them awake at night and their significance and relevance for biomedicine and health. So without much ado, I would like to invite our first speaker, Dr. Dhiraj Kumar. Dhiraj is India Alliance Senior Fellow at ICGEB, New Delhi. So, Dhiraj, if you could, yeah, you there. Okay, fine.
Good morning, and it's really amazing to be here, really wonderful. Thank you, India Alliance, for giving me opportunity to discuss the work that is going on in my lab at ICGEB. This is the place where I work, ICGEB. And the focus of the work in my lab is to understand how, upon infection with Mycobacterium tuberculosis, host responds and takes care of infection. So for the India Alliance fellowship work, I'm focusing on RNA splicing, which is emerging as a new interface between host-pathogen interactions. And I will try to quickly brief through the things that are emerging through this study. While I don't need to introduce you guys with the importance of tuberculosis, the serious problem this disease presents to the public at large. It's a global pandemic, and India contributes really handsomely to the number of tuberculosis patients in the world. And it becomes important to realize that the National Health Mission of Government of India is looking to eradicate tuberculosis. And that's a humongous task, considering that they want to eradicate by 2025, while the global Stop TB program envisions that it should be eradicated by 2030. And there's some major challenges to that, and one of the first serious challenges is to see there are a lot of resistance that is emerging in tuberculosis against the anti-TB drugs. And this is one major problem, that how you can take care of new drug resistance which are emerging in tuberculosis and control the infection. And there are other challenges, of course, like poor vaccines and diagnostics. And these are something, of course, which I'm not going to discuss today. One interesting thing which have emerged in the tuberculosis research, and for that matter, most of these pathogenic infections, is that you start looking to target the host rather than the pathogen itself. And while doing so, it allows you a lot of new avenues to control infections. And I will just brief you with a very, it's like a very terrible accident in the history, but that teaches you about how to look at different diseases differently. And this is an accident in a hospital in Lübeck, Sweden, somewhere in 1930s, where in a neonatal ward, almost 250 kids were accidentally administered with a virulent strain of mycobacteria instead of the vaccine, so which would protect them from disease. And what happened from that is really eye-opening, because what you see that at early, in first 3-4 months, almost 50 kids died of tuberculosis. And as you move forward from there, see some of them developed severe illness, some of them developed moderate, mild, and quite a few of them actually didn't show any symptom. And by the end of 3 years of follow-up, you will see that total number of deaths remained at 70 odd kids, and remaining actually recovered from the illness. What it tells, that host actually plays a very important role in regulating the pathogenesis of tuberculosis. And thereby, if you study host-MTB interactions, you can probably get better avenues to target tuberculosis without targeting the bacteria itself, which has own advantages, because you can target even drug resistance bacteria using these approaches. So very quickly, the host pathogen interactions in case of tuberculosis is complex. That's true with almost all pathogenic diseases. It's multi-layered. When I say multi-layered, it's like it happens at different cellular molecular levels, and it's very dynamic. And to just quickly take you through the very typical tuberculosis infection and follow-up events. So infection generally happens through airways while breathing and taking the bugs, which goes and lands up into alveolar macrophages. And there it goes and affects a lot of early signaling events, including trafficking of the pathogen inside the macrophages, which are the the phagocytic cells, a lot of early signaling events, and metabolism and immune responses are the things which get perturbed. However, the long-term shaping of how the host is going to respond is determined by a change in gene expression profile, and together these events decide whether the bacteria will survive and grow, whether it is going to be persisting or whether it is going to be killed, eventually leading to determine that whether you get an active disease or a persistent disease, or you clearly eradicate the infection. So what this quick review suggests, there are a lot of opportunities. If you know all of these processes, how they are being targeted by the bug, there are a lot of opportunities to target host function, and that will help us achieve bacterial killing without actually touching the bug per se. So among many things that we do in the lab, we look at changes in gene expression to understand how macrophage responds to mycobacterium tuberculosis infections. And if you look into the literature, there are thousands and thousands of papers looking at gene expression changes across different kind of stimuli, whether it is stress or infection and things like that. But what in the literature people have really not gone into is to look at the very interesting aspect of eukaryotic gene expression, which are always expressed in discontinuous stretches of exons and introns. And this can lead to formation of multiple splice variants that brings diversity to the protein complement. It also impacts protein function as well as stoichiometry, its localization to different compartments and interactome. So why we are interested in this? Couple of years back, we did RNA-seq analysis for MTV infected macrophages, and what we found that there are infection and strain-specific alternate splicing event happening in the host. Remember, it's not the bacteria. So when bacteria enters the macrophages, in the host there are massive changes in the pattern of splicing, and that's what was observed here. And the exciting part was that the splicing event had a global footprint. It was affecting almost all those vital pathways which are required to control mycobacterial infections in a cell autonomous way. So to cut really the long story short, the bottom line of this study was that you have a splice variant which is full length, which is purely functional splice variant. If that gets increased in expression, you will see bacterial killing. But then during infection, what happens that you get a truncated splice variant or a variant of a normal protein, and this particular one leads to survival of the bacteria. And without getting any further dramatic, what I'm just trying to convey is that host response tries to make its entire armory, but in the presence of bacteria, they gets broken up. It's in a very literal sense, because the splice variants actually don't form the functional proteins. Many of them actually gets degraded through a lot of processes like nonsense mediated decay and so on. So through this study, what we had established that RNA splicing could certainly emerge as an interesting interface between host and the pathogen. The question was that how bacteria is regulating this very vital function inside the eukaryotic cells, and that's where the India Alliance Fellowship came into the picture. And so our hypothesis was that mycobacterial factors like proteins, different virulent proteins of mycobacterium, can come out into the cell and actually directly regulate a splicing event, considering that RNA splicing is a multi-step process which involves assembly of large multimolecular complexes, and through interacting with some of these proteins or with the splice junctions, you can get alternate splicing regulated by MTV factors. And we had multiple strategies worked out, including high throughput in vitros and candidate approaches. I will very briefly rush through the things that has happened. So we generated some early evidences, like we had a lot of these mycobacterial secretory mutants. So they will be defective in secreting the virulent molecules inside the host cell, and some of those were available in the lab, and some we got from our collaborator next door, Vinay Nandikuri. And we used almost 20 odd, from the RNA-seq data molecules, and looked at what happens to their splicing when we infect the macrophages with one of these mutants. And the common theme was that when you use these secretory mutants, the effect on splicing is less dramatic than what you observe when you have a wild-type virulent strain of Mycobacterium tuberculosis. At the same time, if you just clone these secreted proteins into a eukaryotic expression vector and express into the macrophages, what you see that there is dramatic increase in the alternate spliced variant of the molecules that you were looking at in the RNA-seq data. So this kind of gives you, a stronger indication that yes, mycobacterial proteins are directly playing a role in regulating RNA splicing in the host. But to get to the more direct evidence, we decided to follow in vitro splicing assays. And the way it works is that, if this is the mRNA pool that you want to look for, that how the splicing is happening. So, you can identify some constitutive splicing events. So these two exons, 52 and 74 nucleotide, will always come together no matter which splice variants you are looking at for that particular gene. And then you can clone them to something called mini genes. So that includes the intron also in between these two exons, and they are cloned into an expression vector. And you have a T7 promoter which, helps in vitro transcription to make mRNA, which will be used as template or substrate for doing the in vitro assays. You also have these exon skipping event. This we all identified through the RNA-seq. So what happens here, that you will get two possible splice variants, one where the middle exon is retained and one where it is not retained. This is called exon skipping event. And again, you make the mini gene of that, cloning into the pcDNA3 or something very similar vector like that. And for in vitro splicing assays, what you do is that you first do in vitro transcription of the mini genes. So here is the constitutive, and below is your exon skipping events. You also prepare some the splicing competent nuclear extracts. So these extracts will be able to do the splicing reaction if you provide them the template. So in the presence of the template, you do in vitro splicing reaction. And so when you do in vitro transcription, at that time you use biotin tagged UTP and CTP to label your mRNA with biotin so that you can purify them later on using a streptavidin. And then you do one step RT, and you see the appearance of the splice variant at 146 base pair. It includes some sequences from the primers. So, and also you can do the same thing for these mini genes. And what you see that the 307 base pair is the standard, which happens in a normal condition. But we also included, secretome, that's the culture filtrate proteins of mycobacteria. So these will have all the secreted proteins of MTB. And when you do that, the 307 base pair band just disappears, and you have only the lower one coming up. And there is a difference in their intensity between RA and RV, suggesting that in the presence of mycobacteria, you do see modulation of a splicing event happening in vitro. So again, raising a notch higher the level of evidence that we have that mycobacterial proteins do regulate RNA splicing in the host. But we also observed something more interesting, that other than these 229 base pair, which you can identify based on NCBI GTF file, that what's the length of exons and introns, what are the boundaries, you do get some other variants also. And we have tried to address that also. But what we did is we followed up looking at in vitro splicing for an effect of bacterial secretome on that. So we cloned several mini genes into these vectors, and the general observation has been that we have done in vitro splicing assays with MTB strains and their, and secretome from all the strains that we have. And we have also done splicing reaction using nuclear extracts from MTB infected macrophages. So if there is any protein which is secreted and part of the nuclear extract, they will also show the effects. And now we are doing with the purified MTB proteins. So we are actually expressing around 20, 30 odd MTB proteins and trying to do these assays one by one using one protein at a time. So throughout all these experiments that we have done, the general observation has been, so since we were seeing a lot of these newer size of the bands other than what is reported in NCBI, so we started running tape station because that gives you very precise length of each nucleic acids, and you really see presence of a lot of these splice variant which is coming up. And when we looked at the mini genes, so these are the standard donor sites which are highlighted in red. So donor is at the 3' end of the preceding exon, and acceptor is the 5' end of the subsequent exon. So the green ones are the acceptor sites. So these are the standards one based on which we decided design these mini genes. But if we do the splice site analysis, you find that there are several cryptic non-canonical splice sites, and they becomes activated in the presence of bacterial proteins, suggesting that these could lead to a lot of those non-canonical splice variants also being formed during infections. And we are now trying to establish this with individually purified protein so as to show that which particular MTB proteins, how they interact with the splicing machinery and what kind of products they give rise to. Another aspect of the work was to directly see whether MTB proteins are interacting with the host spliceosome function proteins or not. And for that, we had envisioned to use yeast-2 hybrids and spliceosome pull down and followed by mass spec analysis. And here we had analysis, so I don't need to go through the details of yeast-2 hybrid assays. We all are aware of it. But we have a bait library of around 350 proteins, and this is the bacterial secretome. So bacterial proteins which get secreted. So you make a library of that, and this list was based on 4 or 5 published paper, and we are also doing one secretome assays which has shown some very new proteins from MTB which get secreted. And we use around 30 odd cytosolic proteins as controls to see that whether they also interact with the host spliceosome machinery. For the prey library, we used human splicing genes, which are around 200 odd genes, but we also looked into their splice variants, because what happened in RNA-seq data that splicing variants of spliceosome associated genes were also regulated. So we used around 600 different protein forming transcripts corresponding to these genes to see that and use that in the prey library. And we followed very high throughput gateway cloning to make the bait library and the prey library. So the bait library is ready, and the prey library, considering that these are all different ORFs for these 200 splicing related genes. So they are novel and it's not available anywhere, so we are preparing it in-house. So almost 200 are already ready, 100 is in the topo vector, and another 100 is amplified, which should happen in next couple of months' time. And the interesting improvisation that we did from what we began with is that we decided to do one-to-one interaction using robotics, because initially you would otherwise just put all the baits in one tube and then add one prey at a time and identify the interactors. Here we are doing one-to-one using robotic arms so that you can see that which ones are really interacting or not, and you can select on the on 2 dropout, 3 dropout, and 4 dropout. It's the standard yeast-2 hybrid assays. But coming to the very early results that we have got so far, around 301 MTB secretome, another 40s are not working in this particular strain, so you have to change the strain for that, and around 22 human spliceosome transcripts, and which has led to around 100 or so odd interactions coming up, and the sizes here suggest those which are showing more interactions. So the diamonds are the host proteins and the circles are the MTB proteins, and some of them are showing very nice interactions. And just one example which is very exciting in this, which we came to know later, that these 2 transcripts had very similar interaction patterns from the MTB proteins. And when we looked into that, it turns out they are from the same gene, U2AF1L4, which is part of the U2 accessory factor, which recognizes the 3' splice junction. And these 2 differed in just 2 small stretches of amino acid sequences, and they had amazingly common interactors that are coming up through Y2H and some unique interactors. So of course we are trying to develop this further and understand what's happening. In parallel, we had proposed to do mass spec analysis of in vivo assembled spliceosome complexes, which was very tricky to do initially, and we had planned to do some labeling experiments, although the level of incorporation was very low, so we were not able to find that. But we were able to purify the in vivo assembled spliceosome using anti-Y12 or what is called anti-Smith antibodies, and we have done very early mass spec analysis of that, and we do identify mycobacterial proteins, and 2 of them are present in our Y2H bait library, although none of them have shown interaction so far, but we hope that we will get something very soon. So what has emerged through whatever little bit of work we have done so far is that it's certainly emerging as RNA splicing emerging as a point where MTB interferes with the host functions. Although this is true for MTB, which is emerging now, there is nothing known about other bacterial pathogens which can perturb the host RNA splicing. It's well known in case of viruses, and we reviewed it with our students that how the splicing machinery is regulated under different pathogenic infections. And there are a lot of examples for viruses, but MTB, there are some indications, but there is no direct evidence yet, which I believe that with the more development of this work, we'll get to know how MTP proteins directly regulate that. And probably that will allow us to exploit host RNA splicing as a machinery to target bacteria inside the host. So I will just conclude here. Most of the work has been done by Kumar, who is a PhD student, and Nishat, who is a postdoc in the lab. Harun was the one who started the RNA-seq work in the lab, and Ravi, Raman, and Deepanvita are helping at different stages. And just to highlight what Shahid introduced, that we have trained a lot of these undergrad students. This work allowed me to train these 4 undergrad students who came for short-term training, 2 months to 4 months to 6 months, and they have done amazing work, and when they go back, they are very happy because they could spend a lot of time doing very high throughput works in the lab. And these are the collaborators. Of course, I had to thank Vinay for providing all the knockouts that we have. And Neil will be helping in protein purification. And for mass spec, we have Ben and Rudy. And Ram Yadav at ICGEB Mohali help in doing the robotics, to do Y2H screening, because that makes life very easy. It's difficult for the postdoc to run from Delhi to Chandigarh, but that's all right. Still, it is working. And thank you for your attention. I'm happy to take questions.
Any questions?
Dhiraj, very nice work. The question that I have is regarding the response that you get at the end of the interaction between the host and the pathogen. Is it a cause or a consequence of the variations that are already inbuilt in the host? Because you do have variations in the host,
yes,
which allow varied response.
Yes.
In that sense, what it means is that you could possibly look at population-based difference. That's the direction that one can take.
Excellent point.
Yeah.
And
do you have some comments on that?
Yeah, this is something which I am following in parallel. So what our idea was, in fact, that is briefly discussed in the PLOSPath work. So we had validated our RNA splicing events that we were getting in macrophages, that's the THP1 macrophages, in human donors. We take the human volunteers and looked at it. So what we find that if you infect them with mycobacteria, while the splicing patterns remain similar, but there is still some distinction between different volunteers. So, some splice variants will be more expressed, highly expressed in one volunteer, but not in another one. But there, some other splice variant will change. And the only way you can explain it is having variants at the splice junctions or those cryptic sites, how they are present into the human population. So to look into that, we have now collaborated with a colleague who has generated around RNA-seq data from monocytes from TB patients and healthy individuals. And in parallel, we are supplementing it with whole genome sequencing data so that you can look at splice junction variants. Although this is not part of the Wellcome work, but this is going on in parallel. And we are trying to establish that if you see a splice variant which is coming in the TB patient versus the healthy, can you correlate it with the splice junctions and looking at the context where they are present? So yeah.
So just an additional remark. So different strains also will give you a different response, correct?
Yeah. So
that will be also a bit tricky.
Yeah. So that probably also boils down to different strains will have different secretome. So idea is that we can attack it from all the angles. You look at the secretome and go back and see that whether they are interacting or not. If they are interacting, then of course you have to do one at a time using proteins and express them, and then see whether in vitro whether it is directly affecting the splicing or not. Because there you can be more sure. Here in the cocktail of secretome, you are adding a lot of proteins. But it was early to it's very important to show that even by using these different protein cocktails, you can achieve what you are getting into the in vivo situation. Yes.
Dhiraj, nice talk. One of the things that puzzles me is that you're apparently picking up so many interactions between multiple secreted proteins with components of human spliceosomes. So I wondered whether you saw any shared structural or peptidic motifs that might explain how multiple secretome components were apparently interacting with components of the human spliceosome.
So this is too early because we have only done for 22 praise now. The number has to go up to 450 to 500. But even with this small example, the example of U2AF1L4,
yeah,
it's very clear. They had almost, the entire sequence is similar except for two small stretches of amino acids. and that's what probably is giving to some unique interactors. But around 8 or 10 of those interactions are common between those two splice variants. It actually vindicates our initial idea that we should use each of the protein forming splice variants as prey to look for actual interactors. Other reason we are finding more interactors is because we are doing one-to-one. You know, the problem is all the Y2H so far, when people do it, they club the entire bait, tail labyrinth, add one bait at a time, and then do sequencing to identify which proteins are interacting. Here we are doing one-to-one. Of course, that increases my workload because eventually we have to co-express them into the macrophage system and do pull downs to show that they are interacting in vivo also or not.
No, but of course, it also, though, because you've got highly purified proteins in large quantities, you might also expect to pick up some spurious interactions which won't validate.
Yeah. So, of course, there are at least 3-4 different strategies planned. I mean,
of course.
To just validate all the interaction that we are getting. Yeah.
Very quick question.
Yeah. Did you eliminate rRNA from your analysis?
rRNA? Yeah. rRNA. Yeah, they are not there in the RNA-seq data.
Okay, because that will be interesting to look at the heterogeneity at the ends or whatever.
Right, I know, but I think that has to be done separately because the rRNA, once you get into that, what kind of model? In fact, even the long non-coding RNA data was part of the RNA-seq data, but we couldn't get into that because the splicing itself is so challenging that you can't really spread thin on looking into those link RNA profiles that we generated.
Thank you, Dhiraj. Dhiraj's poster is going to be up there as well. So if you have any other questions, you can catch him at the poster. Thank you, Dhiraj. Thank you once again.